Chapter 9 is all about muscles, and there are lots of them we're going to be learning about. Some of the general functions of muscles include things like muscle tone, moving of bones. Even your heartbeat is based on muscle functions. Every movement that we make requires the use of muscles, even just the expressions we make with our face. Muscles use chemical energy that is stored in nutrients to exert a pulling force on structures that they are attached to. They also help distribute heat, generate that heartbeat, provide muscle tone, and propel body fluids. We're going to focus mostly on skeletal muscles which attach to bones and the skin of the face and under conscious control. And so we're going to look at the three types of muscle tissues. We have skeletal. Skeletal muscle is usually what is attached to bones. It is voluntary. We move these by thinking about moving these for the most part. They are somatic and they are striated. And so we're going to pay attention to the fine details between the muscles so that we can be able to differentiate these in lab. So when we're in lab, we want to really pay attention to the characteristics of the muscle. And actually that's histology, which we're actually probably done with by now. But you still will need to be familiar with those three types of muscles and the way that they look during lecture, especially when we talk about muscle cell contraction. Then we have cardiac muscle, which is unique to the heart. It is involuntary. It is so that it is autonomic. It is also striated. We have smooth muscle, and this is going to be along things like your viscera, your internal organs, blood vessels, and skin, and it is involuntary and autonomic, and this one is not striated. Skeletal muscle has nervous tissue, blood supply, and connective tissues all involved with them, so it is important to pay attention to that. We have coverings over muscles, and this is often what you see as the white portion here in this diagram. We have a fascia which separates and holds the individual skeletal muscles in place, and it is made of connective tissue. We have tendons, which is also connective tissue, and that surrounds each muscle, and it kind of projects past the end of the muscle in a cord-like type structure, and they attach muscle to bone. Aponeuroses are kind of broad, flat sheets, and they are also connective tissue, and they attach muscle to bone or other muscles. You can see an example of these here in this picture at the top here. On the top of the head, we have some aponeurosis here. You can see some tendons here, and these really do have a cord-like structure there. Fascia you cannot see, but you do have fascia like along the bottom of your foot. If you're familiar with like when you are utilizing deer meat or a pork loin, something that still has fascia attached to it, when you pull in, it's kind of a silvery part that you kind of actually have to slide off. That's fascia. We have three types of connective tissue coverings that are involved with muscles: epimysium, perimysium, and endomysium. Epi, remember, means above. So epimysium is going to be the uppermost or outermost layer. And then peri, remember, means around. So it is going to surround something. And then endo means within. So it is going to be the innermost covering here. So epimysium is the outermost layer of connective tissue surrounding a skeletal muscle. And so you can see right here is the epimysium. So we have epimysium covering that. And then there's also some fascia up here. All right, and so the epimysium is going to be the outermost covering. Perimysium is going to extend inward from the epimysium and is going to separate the muscles into small sections. And these sections are going to contain bundles of skeletal muscles that we call skeletal muscle fibers that we call fascicles. And so perimysium, you can see, is right here. And if you will, well, let me get my pen working, is right here. And you can look a little more closely at this diagram in your textbook. Okay, but so perimysium helps to cover that fascicle. And then next we have the, oh wait a minute, before that, each muscle fiber in the fascicle is called a fasciculus, and that is between a layer of connective tissue. All right, so endomysium is going to be the innermost, and that is going to separate individual muscle fibers from each other. All right, and so that's going to surround the muscle fiber cells within the fascicle, and so you can see that is outlined right there. Okay, so I'm not sure how well you can see this picture on your screen, so I would really recommend really looking at this closely at the diagram in your textbook. So skeletal muscle fibers form a single muscle cell. These fibers form from undifferentiated cells. That means they didn't change, that fuse together. And so this is how we get a multinucleated muscle fiber. So there were individual fibers that didn't separate, basically. And so they have multiple nucleus. So just below the cell membrane, which is called the sarcolemma, the sarcoplasm, which is what we call the cytoplasm here. And you can see here we've got sarcolemma is a little bit covered up, but that's going to be right up here, okay? And then here we've got our sarcoplasm. So that's kind of like the cytoplasm. And so these have small oval nuclei in them. They have mitochondria and myofibrils. These are going to be thread-like structures, and they run parallel to each other. Calcium is stored in the sarcoplasm. A sarcomere is best described as a unit within a muscle fiber. We also have transverse tubules and sarcoplasmic reticulum. These are going to be well developed in skeletal and cardiac muscle fibers. Myofibrils, remember myo means muscle, are important in muscle contraction. They're very important. These are made of thick filaments made of myosin. I want you to remember that word because this is going to be really important for skeletal muscle contraction. Myosin, those are the thick filaments, and then actin is the thin filaments. So these alternate and produce these dark and light patterns or striations that we see in the muscle. That repeating pattern of those striations form the sarcomere of each muscle fiber. Myofibrils are basically kind of joined end to end. In the sarcoplasm, you're going to have channels that surround each myofibril. Those are going to run parallel to it, forming what we call that sarcoplasmic reticulum. And you can see this here. You can see that sarcoplasmic reticulum, and you can see these alternating fibrils there. In the sarcoplasm, we've got those that run parallel. We have T-tubules that extend into the sarcoplasm, and those are what house the extracellular fluid. We have a transverse tubule that is between two different enlarged sarcoplasmic reticulums, and we call that a cisternae, and it kind of forms a little triad. This is the cisternae here of the sarcoplasmic reticulum, and then in the middle of those is that little transverse tubule. Together we call those the triad. Striations here have two components, and you can see this much better in this picture where it's blown up a lot better. This works kind of like an accordion, so muscle fibers slide in and out during muscle contraction. Okay, so they're sliding in and out. It's called the sliding filament theory, and this is really important for you to understand. They slide past each other. We have I bands, and I bands are going to be the light bands. Okay, so these are going to be light. These are the lighter colored bands here. I can't get the word out, lighter. So those are the lighter bands. These are going to be made of thin actin filaments, and they're held by direct attachment to the Z lines in the center of the I bands. And these are marked and pulled down here where you can kind of see which are which. Okay, and so I is thin. Our Z lines hold the myosin filaments in place, and those are connected with something called connectin. Sometimes it's called titin. A bands are the dark bands. Okay, A bands are dark bands, and they're made of thick myosin filaments, and those overlap with the actin filaments. They kind of overlap, and you can see up here how those are kind of overlapping. We have proteins that help hold the thick filaments in place, and we call that the M line. We call that the M line. The central region in there is called the H zone. The H zone. Sarcomeres, sarcomere in the area that goes from one is the area that goes from one Z line to the next Z line. So you can see here this is the Z line. It kind of makes a Z, okay? And here's the other Z line. In between that is the H zone here, and this whole section here, that is what the sarcomere is. It's that little section from Z line to Z line. Thick filaments are made of several myosin molecules. And you need to be sure you understand myosin and actin and their relationship in muscles. There are two protein strands. They're kind of twisted together and have these globular heads. So this little section right here is the myosin head. The thin filaments are made of actin, okay? And they have double strands. They're kind of twisted into a helix, and you can see those thin filaments right there. They kind of have a globular shape to them. Troponin and tropomyosin, those are two other really important words. Troponin and tropomyosin are proteins that associate with the actin filaments. And what that means is, you can see here, you've got troponin right here, troponin right here, and then tropomyosin going along. So when they say associate, they're just kind of attached to it, okay? So they're attached to the actin filaments. Troponin has three proteins basically attached. Tropomyosin, it's the more rod-shaped one. It sits in the longitudinal grooves of the actin. Basically, you just need to know that they're kind of attached together. So together, that's what we call the troponin and tropomyosin complex. Myosin that has those little heads, it's kind of like that double hammer on the end, is kind of like when you cock a gun back. And so it catches on the actin and pulls it forward. Okay, so it catches it and it pulls it forward. They basically are grabbing and releasing. And if they're still primed again, they're going to grab and release again, grab and release again, grab and release again. And that brings the muscle closer together.